Okay, for this slide, let's take a look at the directions first. Determine whether each genotype is heterozygous or homozygous. Drag and drop the genotypes into the correct columns. So the first thing we need to know is differentiate the words heterozygous and homozygous. Always remember that homo means same and hetero means different. So when I look at this first one, I see two capital A's, which are both the same. So I add that to my homozygous column. Capital B and little b are different, so I would add that to my different column. And so on and so forth. Big E, little e is different. Big I, little I is different. Big M, little m is different. Little f is same. Big J, little j is different. Big C, little c is different. Big D is the same. And so on and so forth until I finish all of my gametes here. Big H is the same, big L, little l is the same, and here we go. For the next part, it says directions. Drag and drop the phenotypes into the correct boxes. Those are these. You will not use all the phenotypes below. So for the left one, it says purple flowers are big P, and they're dominant to white flowers, which is little p. So when I see a dominant allele, we know that the dominant one will show over the recessive color. So two big P's is the easy one. We have purple flowers, which means two little p's will be white flowers. The harder one is the big P and the little p. We have one dominant allele and one recessive allele. However, because there's one dominant allele, it will always show the dominant color. So go ahead and try it on this side. Again, the easy one is going to be the big R, which is round, which is dominant, and little r, which is recessive, wrinkled. So again, when we have a heterozygous, a different, with one dominant allele, it will always show the dominant color. So here we have our answer.